Hello everyone and welcome back to my channel. My name is Shirsha and today I'm going to be discussing with you about Western blotting. So previously the topics that we have covered so far on this channel include SDS-PAGE, which is a basic tool that is routinely utilized in laboratories everywhere to measure, to visualize and to quantify proteins. And then we also talked about the different types of polyacrylamide gel electrophoresis that are available and what are the benefits and drawbacks of each of them. Next, we're going to move on to discuss about Western blotting. Now, Western blotting is a step ahead of polyacrylamide gel electrophoresis. How so? So, what is Western blotting? The goal of Western blotting, also known as immunoblotting, is to identify with a specific antibody a particular protein which is your antigen within a complex mixture of proteins which have been separated on a SDS page and transferred onto a membrane. So as you can clearly understand from the first line, to do a Western blot, you need to first run an SDS page. So imagine that you are looking for a specific protein. So when you do an SDS page and you stain the gel, you get some bands. But you do not know for a fact if the protein that you are seeing or if the band that you are seeing, is it of the desired protein that you are looking for. So a very good way to confirm that is by Western blotting. Yes, definitely. Your SDS page will give you an idea regarding the size of the protein, and because if you're looking for a specific protein, you would be knowing its size and you can then guess from there if this is your correct protein. But there is no way for knowing for sure. So this is where comes in Western blotting. So what you do in Western blotting is that after you resolved your proteins on the basis of the molecular weight on the gel, you transfer these proteins from the gel onto a membrane. What membrane? We will discuss about it later. And now once your proteins are on the membrane, you probe the membrane with specific antibodies specific for your protein of interest. Now because we all know antigen-antibody interactions are highly specific, so only when your protein of interest is present will your antibody go and bind to it, and then you can visualize the signal. So the individual protein bands are identified how? By flooding the membrane with radio-labeled or enzyme-linked antibodies, which is specific for the protein of interest. Western blotting therefore involves 5 steps. Resolution of the sample on SDS page, transfer of the separated polypeptides to a membrane support, blocking the non-specific sites on the membrane, addition of antibodies, and finally visualization. So the first and foremost step of doing a Western blot is to resolve the sample on an SDS page. So the first step is you take your protein sample, you load it onto the SDS gel, and you let the gel resolve the proteins on the basis of their molecular weight. In case the protein was involved in interaction with some other protein, it would get denatured during sample preparation and it will get separated out in the page. So, for instance, it could be quite possible that your antibody recognizes such an epitope on your protein which is involved in interaction with another protein. So, imagine two proteins are interacting and your epitope of interest lies on the interaction interface. In that case, your antibody can't really detect it. So, typically, we would not do a native page preceding a Western blot. We would always go for an SDS page. Why? Because now the proteins will get denatured and separated from one another. And now this interaction interface is free. And this interface contains your epitope. So now your antibody can come and bind to it and recognize it. The next step of a Western blot is to transfer the separated polypeptides onto a membrane support. Now this step is very crucial. So after being separated by SDS page, these antigens are transferred or immobilized onto a membrane. Now there are typically two types of membranes commonly used in Western blotting. You have your PVDF membrane or you have your nitrocellulose membrane. Now these membranes, they bind to proteins very non-specifically. So you have to be very careful when dealing with a membrane. Always wear gloves, because if you accidentally touch the membrane with your fingertips, then that will actually end up showing some black marks when you finally visualize the blot. Okay, also always use a pair of forceps to ensure that touching of the membrane is minimum and least surface area on the membrane is touched. Now, nitrocellulose membranes are popular matrix used in protein blotting because of their high protein binding affinity and compatibility with a variety of detection methods. Protein immobilization is thought to occur by hydrophobic interactions, and high salt and optimal methanol concentrations help improve this protein immobilization. So during the transfer process, some buffers are added, and these transfer buffers always contain methanol in it because methanol helps with this immobilization. PVDF membranes, which is the second choice, are also highly hydrophobic and must be pre-wetted with methanol or 95% ethanol prior to submersion in transfer buffer. If you forget to pre-wet your membrane, then you can forget about any transfer occurring. So it is very crucial that you allow it to soak in methanol or 95% ethanol for at least 30 seconds, and then you transfer the membrane onto the transfer buffer. Now, PVDF membranes also have a very high binding affinity. Binding likely occurs via dipole and hydrophobic interactions. Now, both varieties of membrane are chosen for their non-specific protein binding properties and must not be allowed to dry out once placed in the cassette. Now, it is very important to keep in mind that a membrane should not go dry during the entire procedure, and membranes are selected for their ability to bind to proteins non-specifically. So that is why you have to be careful with the membranes when dealing with them, because if you accidentally touch the membrane in the exact same region where your protein of interest was supposed to show up, then it will obviously obscure your signal, and then you would not get any proper conclusive results. So transfer is usually achieved by placing the membranes in direct contact with the gel and placing this sandwich in an electric field to drive the proteins from the gel to the membrane. So for transfer, we once again utilize the fact that the gel that we had run to resolve these proteins was an SDS-PAGE. So your protein is already coated with SDS, it has a net negative charge, and if you place it in an electric field, it will migrate towards the positively charged electrode. So this feature is taken advantage of, and transfer is also achieved by applying an electric current. The gel and the membrane are placed in direct contact with each other, and you apply an electric current such that the membrane is facing towards the positively charged electrode, and so once your proteins move out of the gel, it will move on to the membrane and it will get immobilized or captured there. The proteins will not move out of the membrane. So typically PVDF membrane comes in the form of rolls as shown over here, and nitrocellulose membranes would also come in similar rolls. So freshly electrophoretic SDS gels are laid flat on pre-wet membranes, which are supported on layers of filter paper that are wet with a transfer buffer. So why do we add these layers of filter paper? First reason is that in case you have any particles or anything in your transfer buffer, to sort of filter that out, and also to create an overall flow, to create an overall flow of ions through the entire setup, because you don't really, unlike when you were running the gel, your gel was directly immersed in the buffer. In case of transfer, we will see shortly, you may not always do that. So you need a uniform flow of current around, all around this entire setup, and this is where these pre-wet filter papers come in. So the gel is overlaid with filter paper wet with transfer buffer. So basically you have filter papers on either side. Now care should be taken to exclude any bubbles between the gel and membrane, and between the membrane and the paper. So if you have a bubble between your gel and membrane, in that case this air bubble will hinder the migration of proteins from the gel onto the membrane, and when you develop your or try to visualize your blot, you will clearly see a bubble in place of where the bubble actually was. So yes, you should always take care to exclude bubbles. The whole assembly is then locked together in the form of a cassette, but this cassette is only used for wet transfer. In case of the other kind of transfer, the semi-dry transfer, no cassettes are used. The transfer buffer usually contains tris and glycine dissolved in an aqueous solution of methanol. So tris and glycine is very similar to the buffer that was used in case of your SDS polyacrylamide gel. pH values used are above 7 in order to ensure that all of the proteins migrate towards the anode. And methanol is very necessary to prevent gel swelling with heating because a lot of heat is generated within the transfer system during the process of transfer. So methanol acts as a cooling agent which prevents the gel from swelling up. And this in turn improves protein immobilization onto the membrane. So proteins move from within the gel onto the membrane while maintaining the organization that they had in the gel. So this is very, very critical to keep in mind that the organization, the relative orientation, the relative arrangement of the proteins are not at all affected by this entire transfer process. The transfer is cathode to anode because the proteins are negatively charged. They're coated with the negatively charged SDS molecules. So the membrane must always remain on the anode side. So take care of that. So this is what a typical transfer setup looks like. So you have your filter paper towards the cathode, then you have your gel, you have the membrane, and then you have more filter papers on the anode side. So over here, only one filter paper is shown for representative purpose, but typically more than one are used. So coming to the different kinds of electrophoretic transfers, there are two kinds. First is the wet transfer. So this wet transfer procedure involves a complete immersion of a gel membrane sandwich in a tank containing transfer buffer. The electrodes are kept vertical. These are the graphite, stainless steel or platinum electrodes. Tank systems are typically run at a constant voltage. Mixing of the buffer during transfer keeps the current relatively constant. So this is what a wet transfer setup looks like. So if you can recall, in the previous slide we were talking about cassettes in which the entire gel membrane filter sandwich is locked. So this is what the cassettes typically look like. And this entire transfer tub looks very similar to the tubs that we use for polyacrylamide gel electrophoresis. So you lock your sandwich in the cassette and then you immerse the sandwich in this transfer tub. And the tub is filled with buffer. So this actually acts as a helps in maintaining a continuous medium of current. And this is how your wet transfer goes on. And typically, wet transfers are carried out for longer periods of time. And because they're carried out for longer periods of time, it's typically carried out at lower temperatures, say roughly overnight at 4 degrees Celsius. Next, we have semi-dry transfer. So this involves placing the gel membrane sandwich between absorbent paper, which is soaked in the transfer buffer to help maintain the medium. The electrodes are plates of stainless steel or graphite. So here the electrodes are these vertical rods, but in case of semi-dry transfer, they are plates. This technique is as effective as wet transfer and far quicker. Within 15 to 45 minutes, it can be completed. However, semi-dry blotting systems have lower buffering capacity and therefore they're inappropriate for prolonged transfer. It is quite natural because unlike in wet transfer, there is not a continuous supply of buffer. You soak your sandwiches in the buffer and whatever buffer the sandwiches have soaked up, that is all you have available. So you have to keep it in mind that you cannot allow this semi-dry transfer to go on for too long. Then it will dry out the entire system. So this is what a typical semi-dry transfer machine looks like. So basically on this plate you place your sandwich, the gel membrane filter paper sandwich, and on top of it you place this other electrode. So you see both the electrodes are like flat stainless steel plates and here you have these rods. So the next or the third step is blocking of non-specific sites on the membrane. So before the blot can be processed for antigen detection, it is essential to block the membrane to prevent non-specific adsorption of antibodies. Because you have to keep in mind here that antibodies are nothing but proteins, and because these membranes bind to proteins non-specifically, so whatever region of the membrane that is not occupied by the antigens or the proteins that have been transferred from the gel onto the membrane, so these regions, they are still empty. They can bind to any protein non-specifically. So it is very, very crucial to block these sites on the membrane to avoid non-specific binding of the antibodies to these sites. So blocking of non-specific binding is achieved by placing the membrane in a solution of typically 1 to 3% BSA, which is bovine serum albumin, or non-fat dry milk. in a buffer with a small percentage of detergent. Why the detergent? Because that helps in ensuring that binding has certain level of specificity and that nothing is, these um, BSA or these non-fat dry milk molecules are not just binding haphazardly or randomly. The protein attaches to the membrane in places where the target proteins have not attached to prevent further non-specific binding of proteins. So this is what like it's a very generic image showing that you have this container with your blot after transfer, and this experimentalist here is pouring in non-fat dry milk, skimmed milk basically. That's what we use onto the membrane, and then you incubate the membrane with this either BSA or skim milk for say roughly 1 hour at room temperature, and that is enough to block all non-specific sites. If you are using freshly prepared BSA, then you can use even shorter time periods of incubation, like say 30 to 45 minutes even. So step 4 of the Western blotting procedure is addition of antibodies. So you have run your gel, you have transferred the resolved proteins onto the membrane, you have blocked the non-specific sites on the membrane with some inert protein like BSA or skimmed milk. Now you will be adding the antibodies specific for the proteins you want to detect. So virtually all Western blots are probed in 2 stages. An unlabeled antibody specific for the target protein of interest is first incubated with the membrane. The membrane is then washed to remove any antibody that is sticking to the membrane non-specifically and incubated with a secondary antibody. Now, the secondary antibody is an anti-immunoglobulin that carries a reporter group which is either radiolabeled or coupled to an enzyme. One-step detection methods, although more expensive and less common, are also available. So this method is a two-step detection method where you incubate your blot with 2 antibodies. The first or the primary antibody was specific for your protein of interest, and the secondary antibody is specific for the primary antibody. The secondary antibody is the one which carries the reporter group to help with visualization. One-step detection methods are also available wherein you will be incubating your blot with only 1 antibody, but this is more expensive and therefore less common. So this is your one-step detection where you have this antigen or protein immobilized on the blot and you're adding your antibody which is conjugated with a reporter group. So this is direct detection. Now, one of the possible drawbacks with direct detection can be when your antibody is getting labeled, then the paratope of the antibody, which is essentially the epitope recognizing part of the antibody, might also get labeled. And if these, like for instance, these molecules, and this labeling will actually interfere with binding of the antibody with its specific epitope. And then you have indirect detection, wherein you have your epitope or your antigen of interest and your primary antibody specific to it, which binds to it. And now you have your secondary antibody specific to the primary antibody, and the secondary antibody carries the reporter group. So two-step detection, also known as the indirect method of detection. So after blocking, a solution of primary antibody, typically at a dilution of 1:500 to 1:2000, is incubated with the membrane under gentle agitation. So you typically keep your blot in a box and you add this antibody solution and you keep it on the rocker. The antibody solution and the membrane can be incubated together for any period of time between 30 minutes to overnight at varying temperatures. So what temperature you're incubating at plays a crucial role on determining how long the incubation period is. So if you're going to incubate for say 30 minutes to 1 hour, you would typically incubate at room temperature. But if you're going to incubate at 4 degrees Celsius, then you incubate it overnight. Also, it also depends on the affinity of the antibody for your protein of interest. Not all antibodies have a similar affinity range. Higher affinity antibodies can be incubated for shorter periods of time, but there are some primary antibodies which do not have so much high affinity, and over there you typically incubate it overnight at 4 degrees Celsius. Please ensure that you do not leave your blot overnight at room temperature because that will not be good for it. After washing the membrane to remove the unbound primary antibody, the membrane is then exposed to a second antibody, which is your secondary antibody, which is an anti-immunoglobulin coupled to a reporter group. Typically, the secondary antibody is an enzyme conjugated, or it can also be radio-labeled, anti-immunoglobulin, consisting of an antibody against a species-specific portion of the antibodies being tested for. For example, if mouse anti-HIV antibodies are being used for the assay, then the antibodies, which will be the secondary antibody, is something that is raised in a different species, say for instance goat or rabbit, and is specific against mouse immunoglobulins, and these are conjugated to the enzyme. Due to its targeting properties, secondary antibody tends to be therefore referred as anti-mouse in this case, or anti-goat, anti-rabbit, etc. So yes, if you take a look at this, you would see that you have your protein immobilized on the blot. You have the primary antibody against it, and then you have the secondary antibody. So in case you don't know, or even if you know, let's just brush up briefly. An antibody has two parts. This is the Fc portion, which is the constant region, and this is the Fab portion, which is the variable region. The Fab portion comprises of the regions or the variable regions that are specifically able to recognize specific regions on the antigen. And this Fc region is conserved. So because the Fc region is conserved, the secondary antibody used is always specific against the Fc region. So for instance, for example, you can buy just say all of your antibodies which have been raised in rabbit, and then you would need just one secondary antibody, the anti-rabbit antibody, to detect this entire range of rabbit antibodies. Why? Because the Fc portion would be common. And your secondary antibody is conjugated to an enzyme, say. So when you add the substrate, there'll be a reaction and then you would get a signal. The other kind of detection is one-step detection or the direct method of detection. So one-step probing systems require a probe antibody which both recognizes a protein of interest and contains a detectable label. So one antibody is doing both the jobs. It is both recognizing the immobilized protein and at the same time it has the enzyme conjugated to it. The primary probe is incubated with a membrane in a manner similar to that for the primary antibody, and this is then ready for direct detection after a series of wash steps. So in two-step detection, after primary antibody incubation, you would wash the blot and then you would incubate with secondary antibody. But in case of one-step detection, you directly develop the blot after incubation with the primary antibody and washing. So this is what it's like. So now you see your primary antibody here has the enzyme conjugated to the Fc portion, and when you add the substrate, the subsequently the reaction ensues and you get the signal. So coming to direct versus indirect detection method, both of them have their advantages as well as disadvantages. So let us go through some of them briefly. So in terms of time, obviously in case of direct detection method, the major advantage is that you can develop your blot after only one step incubation. So it's much quicker compared to indirect detection, where the main disadvantage is that the addition of the secondary antibody leads to an extra step. But coming to the disadvantage of direct detection, now all of your primary antibodies have to be labeled. So individually labeling these primary antibodies is time consuming. But if we are being practical, because we are the end users and we are not the manufacturers, so this step does not really affect us much. And the main advantage of indirect detection is that many reliable commercially available label secondaries are available, which reduce the time consuming optimization steps. But again, I would like to say from a practical point of view that you have commercially reliable, well-tested, labeled primary antibodies available for direct detection. But what's the major drawback there? The major problem is cost. We will come to that. So in terms of flexibility and availability, yes, the number of conjugated primary antibodies is limited. You don't really have primary antibodies against every protein that you can think of. But again, more and more with the passage of time, more and more antibodies or primary labeled antibodies are being commercially produced, specific against a wide range of proteins. So there is no advantage here. In case of indirect detection method, the advantage in terms of flexibility and availability is that, like we have discussed, you can have one secondary antibody specific to a certain species, say, and if your primary antibodies, say for example you have 20 different primary antibodies, and if they're all raised in the same species, then say rabbit, then this one secondary antibody, which is anti-rabbit, can be used to detect all of them. So in terms of flexibility, it's super high. In case of signal enhancement option, the disadvantage of direct detection method is sample concentration may be required for low abundance proteins prior to loading. So if your protein is occurring at a low concentration, then detection might become problematic with direct antibodies. But this is the advantage of indirect detection method, because there is a second antibody being added, so that sort of amplifies the signal and it helps you to detect even low abundance proteins. Coming to sensitivity, in case of direct detection method, like we discussed, the signal from direct method may appear weaker than indirect method, and that is the advantage of indirect detection, where you get signal amplification, because each primary antibody can accommodate multiple secondary antibodies on it. So in case of direct detection, you have only one primary antibody binding to your protein, and that's it. That's giving you the signal. So if your protein concentration is low, the number of primary antibodies that bind to it will also be low. So the net signal you will get is less. But in case of indirect detection, because multiple secondary antibodies can bind to a single primary antibody, so even if one primary antibody is binding to your protein band, because more than one secondary antibodies can bind to this bound primary antibody, so that basically amplifies and intensifies the signal, and therefore you can detect proteins which are of low abundance. In terms of immunoreactivity, so the disadvantage with direct detection method is that primary antibodies may have reduced immunoreactivity, as we discussed in a couple of slides back, because the conjugate might interfere with the antigen binding. We discussed it that the antigen binding interface or the paratope of the antibody might contain the conjugated label, and this will interfere with this binding. But no such problem is observed in case of indirect detection method. Because why? The primary antibody is free from any conjugates, so it can readily bind to the protein immobilized on the blot, and then you add your secondary antibodies onto it. Coming in terms of cost, yes, the major disadvantage with direct detection method is that it is really expensive, because you have to buy these primary antibodies conjugated with a label, and they are pretty expensive. Whereas the major advantage of indirect detection method is that it's cheap. Like already discussed, you can have one secondary antibody and you can use it to probe a lot of different primary antibodies. And these unlabeled primary antibodies are much cheaper compared to the labeled primary antibodies. Coming to the final step of Western blotting, visualization. So after the unbound probes are washed away, the Western blot is ready for detection of the probes that are labeled and bound to the protein of interest. So the first method of visualization that we are going to discuss about is autoradiography. So keep in mind, autoradiography was very popular back in the day, but now it is less and less common, because first of all, dealing with radioactive materials is hazardous, and secondly, you need special permissions for that, you need special rooms for that, and much more efficient and simpler ways are now available. So I-125 labeled protein A and autoradiography can be used to detect the bound primary antibody. Autoradiographic plates are placed directly against the Western blot. So yes, your antibody has to be labeled. So we are talking about protein A here. Why? Because protein A, which is obtained from Staphylococcus aureus, binds to the Fc region of antibodies non-specifically. So you can use I-125 label protein A. So you have your blot, which is basically the membrane on which your protein has been immobilized, and you hold the autoradiographic plates directly against it. The plate develops dark regions corresponding to the protein bands of interest. This method is quite sensitive, but its importance, as I already mentioned, is declining, as other methods have been developed which are of similar sensitivity and avoid the hazards of radioactivity. So this is what a typical blot developed by autoradiography looks like. As you can see, you have these bands here. These bands correspond to your proteins of interest. So some bands you see are of higher intensity compared to others. So that means that this protein was present in a higher or greater amount compared to this. Some bands you see are more fuzzy or more diffuse, and this essentially represents the migration pattern of that protein on the gel. The next method of visualization is colorimetric detection. So the colorimetric detection method depends on incubation of the Western blot with a substrate that reacts with the reporter enzyme coupled with the secondary antibody. After binding of the enzyme antibody conjugate, the addition of a chromogenic substrate. A chromogenic substrate is one which produces color. See, so it produces a highly colored and insoluble product that causes the appearance of a colored band at the site of the target antigen. The development of the blot is then stopped by washing away excess of the soluble substrate. So you have to quench the reaction. Substrates used usually include NBT-BCIP or for alkaline phosphate coupled secondary antibodies. So if your antibody is coupled to alkaline phosphatase, then NBT-BCIP is a commonly used chromogenic substrate. Or if it's coupled to horseradish peroxidase, then DAB and H2O2 is a commonly used chromogenic substrate. So this is what a blot developed by colorimetric detection typically looks like. It is not relevant to us what these labels signify, so I'm not going to discuss that. So as you see over here, you see, so the colored insoluble product that was produced as a result of the reaction of the enzyme with the substrate, this insoluble product precipitates in the specific region, and this gives you colored bands. So you see the band itself is colored. Unlike in the previous case, so the band appeared as simply black regions, you get beautiful colored bands with colorimetric detection. The third kind of visualization method is chemiluminescence, and I think chemiluminescence is the most commonly used visualization method, and I for one can say that I use it on a daily basis in my day-to-day laboratory experiments. So chemiluminescent detection methods depend on incubation of the Western blot with a substrate that will luminesce, that is, that will produce light when exposed to the reporter enzyme, typically horseradish peroxidase, coupled with the secondary antibody. So a luxogenic or light generating substrate is used, which takes place of the chromogenic substrate in the previous method. For example, oxidation of the compound luminol by H2O2 and the enzyme horseradish peroxidase, or commonly abbreviated as HRP, produces light. The light is then detected by photographic film, which is developed to visualize the band. Now, in case of chemiluminescent detector, this light is not detected typically by a photographic film, but previously it used to be, but these days a photographic film is not used to detect the light, but rather a camera is used to detect this light produced. The enhanced chemiluminescent or ECL detection is also available, and this is considered to be among the most sensitive detection methods for blotting analysis. So you have your common enhancer peroxidase substrate which you add to develop your blots. You have these other more sensitive reagents that are also available, which if you add to it can detect even the slightest of signals. So this is typically what a blot developed by chemiluminescence looks like. So you see they have titrated the amount of protein present over here, and you can see as they go down, the intensity of the band also goes down. Finally, we have fluorescence-based detection method available, and this is a new and upcoming detection method. It is not as commonly used as chemiluminescence-based methods, but it has some of its advantages which makes it more favorable. Now, fluorescence-based detection method depends on incubation of the Western blot with an antibody, either primary or secondary, conjugated to a fluorophore. So what's a fluorophore? It's simply a molecule which, when excited with light of a particular wavelength, will emit light of a different wavelength, typically higher wavelength, which lies in the visible range, and therefore you can detect you can detect light in the non-visual range as well, but you can visualize it with your own eyes. Excitation of the bound fluorophore conjugated antibody with light of correct wavelength would give a signal. So the signal obtained would be very, very specific. Why? Because unlike in the previous methods, you are controlling which fluorophore is going to give you the light. So in the previous methods, you add the substrate, and wherever the antibody conjugated to the enzyme is available, you will get a band. But over here, because the signal production is dependent on what wavelength of light you're exciting it with, so this is even more specific, so to say. So signal can be detected using suitable digital imaging systems, and this method allows the detection of multiple proteins simultaneously and consequently is increasing in popularity. So this is what typically a blot developed by fluorescence-based detection looks like. So you see the marker proteins are also coupled to fluorophores to allow for their visualization. And because you would obtain signal, like if your antibodies used are conjugated to different fluorophores, then you can visualize multiple signals. Like in this case, like 3 protein signals are visualized: this green one, the blue one, and the red one. So this method of detection is not possible in any of the previous methods that we have covered. Over there, you can detect only one protein of interest at a time, but in this method, you can detect more than multiple proteins of interest simultaneously. But of course, you have to ensure that the fluorophore that you use, their excitation range is close by so that they can be used for simultaneous detection, and that their emissions are of different wavelengths, so you can visualize them as different colored bands. So one last thing I would like to cover with you is the Ponshu S stain. The Ponshu S is a sodium salt of a diazo dye of light red color that may be used to prepare a stain for rapid reversible detection of protein bands on either nitrocellulose or PVDF membranes. Now this stain is useful because it does not have any deleterious effect on the sequencing of blotted peptides, and therefore it is one method of choice for locating polypeptides on Western blots for blot sequencing. It is also easily reversed with water washes, so which allows subsequent immunological detection, that is antibody-based detection. The stain can be completely removed from the protein bands by continued washing. Its common stain formulations include 0.1% weight by volume Ponshu S in 5% acetic acid, or 2% weight by volume Ponshu S in 30% trichloroacetic acid and 30% sulfosalicylic acid. So this is what a typical Ponshu stained blot looks like. It has this light red color, and you can see that the protein bands are easily visualized. So what can be an application of this staining method? Say for instance, you want to visualize multiple protein bands on your blot, but you do not have antibodies against all of them. You have antibodies specific for only one of them. And say for instance, you know that maybe you did some experimentation, so you expect. So now this is something we will cover in the next lecture about immunoprecipitation. But just to give you a brief idea, you did an experiment to assay the interaction of two proteins, say, so protein A and protein B. And now you know you are basically pulling down protein A out of solution, and you want to see if protein B was interacting with protein A and if it is also coming out of solution. So you run the gel and you use an antibody against protein B because that is what you have available to see if it's present or not. but you do not have any antibody for protein A. But you know it should be there because that is the end from which you are pulling the complex out of solution. So what can be one way? You simply Ponchu stain the blood. And because you know the specific molecular weight of protein A, if you get a band at that particular molecular weight, then you know for sure that yes, protein A is here. You expect protein A to be there, but this is just a way of confirming that yes, indeed, protein A was present in it. So this is one application of Ponchu staining. So coming to comparison of different detection techniques. So at the level of substrates, in case of fluorescent detection, fluorescent dyes and conjugated secondary antibodies are required. For chemiluminescence, you need these peroxidase substrates. For chromogenic detection, you need color detection reagents. Coming to detection, so fluorescent detection, you require proper imaging instruments with appropriate filters and lasers. For chemiluminescent detection, you need a film or more commonly a developer system. And for chromogenic detection, no instrumentation is required. You simply take the blot, you add the substrate, you let the reaction go on, and you wash away the excess of the substrate. And now you have a blot which has this beautiful colored bands. So coming to signal duration, fluorescent detection has the longest signal duration. Not longest, the second longest. It can retain the signal for weeks. Chemiluminescent detection has the shortest signal duration of only a couple of hours. And chromogenic detection, well, it has the highest signal duration of months. So you can develop the blot once again after a month and you will get the signal. Some things to keep in mind. Fluorescent detection can simultaneously detect multiple proteins on the same blot, and therefore it is increasing more and more in popularity because it allows the detection of multiple proteins simultaneously. Chemiluminescent detection has the advantage of excellent sensitivity with a wide range of substrates that are available. Chromogenic detection has the advantage of direct visualization with no imaging or film processing instrumentation required. So this is quite similar to how you stain an SDS-PAGE. You take the page, you add your stain, and you get the page which is stained. So you don't need any visualization system. You don't need any specific highly sophisticated equipments which you would need in case of these two, or you don't need any film processing which would be involved in case of chemiluminescent detection. It's just direct visualization of a stained blot. So coming to the applications of Western blot, the applications of Western blot are actually multifaceted. Western blotting can be used to determine a number of important characteristics of proteins or to identify the presence of a specific protein in a complex mixture of proteins. Say for instance, if you have two or more proteins which have the same molecular weight. So when you run the SDS-PAGE, you will not be able to resolve them. So one way to resolve them would be by 2D gel electrophoresis, but that is again more cumbersome, it's more difficult, results are always not so good. So a quick way around is to do a Western blot. If you have antibodies against say these three proteins who have the same molecular weight and therefore migrate at the same rate in the gel, you can simply do a blot and you can just add one by one the antibody solution specific for each of these three different proteins, and you can detect all of their presence or absence as the case might be. It can also be used to determine the quantity of protein, because if you remember the example blot that we saw in case of chemiluminescence, with the difference in the quantity of protein, the intensity of the signal varies. It can be used to determine the relative molecular weight of the polypeptide from the standard marker that you run along with it, and also the efficiency of extraction of antigen. Now that is basically like the immunoprecipitation that I briefly discussed about, and it is something we will cover in much more details in the next lecture. Now, Western blotting can also be used to identify a specific antibody in a mixture. Western blot can also be used for a number of medical and diagnostic applications, including confirmatory tests for HIV and for bovine spongiform encephalopathy or BSE, which is commonly also referred to as mad cow disease. So this brings us to the end of a very detailed and long lecture. I think this is the longest lecture I have posted so far, but there were so many different things to discuss and so many snippets of information from my daily experience that I wanted to share with you. I hope I have done justice to this topic. It is one of the most commonly and routinely used methodologies in laboratories, and it is something that is very, very commonly used. So I hope I have been able to convey whatever I did want to. And in case you have any questions or any queries, please feel free to leave a comment and I will surely get back to you. If you like this video, do like it, share and subscribe for more such videos. Thank you for your time and I'll see you next time.